So in this video, I'm going to talk about protein synthesis, which another term for protein synthesis is just the making of protein, okay, or formation of protein within the cell. Now we know that ribosomes are the main organelle involved in production of protein, but the instructions for producing protein are actually found in the DNA, which we know is housed in the nucleus. Okay, now specifically, we have parts of DNA called genes. These are the parts of the DNA that are going to be actually involved in the production of protein. Okay, so those are going to be the specific segments of DNA that code for proteins. Now, we know that we have many different types of proteins in the human body. Some of the proteins are what we call fibrous, which means they're kind of like fibers. They're like string-like material, rope-like material. Those are typically used for forming a structure within a cell, okay, so helping form a framework within a cell. Globular proteins are the functional proteins. These are ones that actually have some type of function. They could be enzymes, they could be used as hormones, okay, so could have many different functions, but these are going to have more of a three-dimensional shape versus a string or a rope-like shape. And again, they're going to have a more specific function other than just helping to form a structure within the cell. Now again, I told you guys that DNA is basically the blueprint for protein synthesis, okay? And DNA is coded in what we call triplets. So let me just come up with a fake piece of DNA here. So remember, if you have a strand of DNA, remember it's double-stranded, right? So what you really would have would look something like this. If you have the complementary base pairs, remember that A's pair with T's and G's pair with C's. So this is what a double-stranded piece of DNA might look like, okay? Well, one of these strands, we'll talk about this more in a second, is called a template strand. So that's the one that will actually be used during the production of protein, okay? And when you look at that template strand, so let me just kind of highlight it here. For every 3 nucleotides that we have, this is called a triplet, okay? Triplets, 3 babies, right? So every 3 nucleotides is called a triplet. Now, every triplet codes for 1 amino acid. And amino acids are the building blocks of protein. Okay, so basically you take all these little amino acids that we're coding for and you string them together and you end up getting a nice complex protein, okay, which is helping make up part of the body. Now, each one of these triplets is going to code for a different type of protein. So this ACG here is going to code for a different type of protein than this TCA, okay? So keep that in mind. It's going to code for a different amino acid, okay? So ACG codes for one amino acid. This TCA codes for a different amino acid. And then you can string these 2 amino acids together and form a more complex protein, okay? Now again, the organelles that are going to be stringing these these pieces of material together are going to be the ribosome, okay? So ribosomes are the organelles that help produce protein by stringing together amino acids, okay? Remember, ribosomes are found in 2 places in the cell. They could either be scattered in the cytoplasm of the cell, or sometimes they're found attached to the rough ER. But either way, their main purpose is to build protein. Now, keep in mind that when we get ready to build the protein, DNA actually never leaves the nucleus, okay? It has to stay in the nucleus. That's where it's safe. If it leaves the nucleus, it could possibly be damaged, which would be deadly for the cell. So the DNA stays in the nucleus, and what happens is the cell produces a copy of the DNA in the form of RNA, okay? So it takes DNA and then it converts it into RNA, ribonucleic acid, which it will then send out of the nucleus into the cytoplasm. And then that RNA can be used as the instruction template for the production of protein. Okay, so before we can produce protein, we got to make a copy of our DNA in the form of RNA. So we got to convert our DNA to RNA. Now, what are some differences between DNA and RNA? There's three big ones that I want you to be familiar with. Okay, for one, DNA is a double helix structure. Okay, it's double stranded. So here's a picture of DNA here. Again, it has two strands that are stuck together and then twisted up. Okay, so DNA is double stranded. RNA here on the left is single stranded. It's just one strand that's twisted. Okay. Now with DNA, another difference is the sugar that's found in the backbone of the DNA. Okay. DNA contains a sugar called deoxyribose, hence the name deoxyribonucleic acid. Okay, so DNA contains deoxyribose. RNA contains the sugar ribose. So there's a different sugar found in the backbone, okay, between these two. And the third major difference is DNA contains the base pairs C, G, A, and T, whereas RNA contains C, G, A, and U, okay? So there's one base pair difference between DNA and RNA. DNA contains the thymine, or the T, whereas RNA contains a different base pair called uracil, or U, okay? So those are going to be the three big differences between these two structures. So again, the DNA is found in the nucleus of the cell, right? We use the DNA to make a copy in the form of RNA, and then we can send the RNA out of the nucleus into the cytoplasm, and the ribosomes can use that RNA as the blueprint for the production of protein. Now we have three different types of RNA that we'll talk about. The one that actually is produced within the nucleus and then sent out to become the blueprint for the production of protein, that specific type of RNA is called messenger RNA. It helps make a message and carries that message out of the nucleus and into the cytoplasm. Again, the message is the instruction for the production of protein. Okay, so that's the big one we're going to be talking about. Now we also have ribosomal RNA. This type of RNA is found in your ribosomes. Okay, so it helps make up the ribosomes, which we know, again, those organelles are used for production of protein. Okay, and then we also have transfer RNA, or tRNA. These are going to be used to help add amino acids to our building, to the protein that we're building. Okay, so these are going to actually carry in the amino acids that need to be added on or linked into our protein to help make our protein bigger and more complex. Okay, so tRNAs are going to be carrying amino acids and helping build build the protein by linking together those amino acids. Ribosomal RNA is the RNA found in your ribosomes, which are the organelles used for protein production. And then messenger RNA, again, is the RNA that's produced inside of the nucleus by copying the DNA, and it's used as an instruction template to get sent out for the ribosomes to read in order to produce protein. Okay? So let's talk about the process of protein synthesis. Now, there are two major steps, okay, within protein synthesis. There is transcription and translation. Now, transcription, okay, is the process. Let me just write up here. So transcription is going to happen inside of the nucleus. And this is where we take our DNA and we convert it into RNA, more specifically messenger RNA. Okay? So that's going to happen inside the nucleus because remember, DNA never leaves the nucleus. Okay? Now translation is the second step. So what happens here is we take this mRNA and we send it out of the nucleus, out into the cytoplasm. So this is going to happen out in the cell cytoplasm. And we convert that mRNA into protein. Okay, so transcription happens in the nucleus. That's where we make mRNA from our DNA. And then we're going to take that mRNA and send it out into the cytoplasm of the cell. And there we're going to use it to produce protein. Okay, so that step is called translation. So those are the two big steps, steps 1 and 2 in protein synthesis. Make sure you're familiar with that, okay? Now this is a really busy slide, so I'm going to try to break this down for you as best as I can. Again, I said transcription, okay, that's step 1 in protein synthesis, is going to happen inside of the nucleus. So in this big picture here on the right, the nucleus is up here in the corner. All of this in purple is the nucleus. So you can see your double-stranded DNA, right? And you can see it's being unzipped, it's being pulled apart. So you can see the two purple strands here being pulled apart. This blue strand that's being formed is going to be our RNA. Okay, so that's what we're making during this process of transcription. So basically you unzip the DNA and then you add base pairs onto the already existing DNA strand. Okay, now keep in mind that there's a base pair difference between RNA and DNA. Remember, instead of having T like we have in DNA, we're going to add a U instead. Okay, when we're building our RNA strand. So for example, if you had a strand that looked like this, right, of DNA, your RNA strand would actually be UGCA. Okay, so we would replace our T with a U. So keep that in mind as you are looking at production of RNA. Okay, if I were to ask you a question on a test and say, you know, what would be the RNA strand produced from this DNA strand, make sure that you include the uracil, the U, instead of the T, right? If you're talking about RNA, that's a big difference between those two. Okay, again, that process is going to happen in the nucleus. So we're just building an RNA strand. Here's our mRNA from our DNA strand. So the one strand that we're using, remember I said this is going to be our template strand. So that's our instruction template. And then we just add bases onto that to get our strand of mRNA. So once we're done making our mRNA, it kind of breaks off of the DNA, and then it can travel out of the nucleus, okay, where it can be converted into protein, made into protein. So whenever I told you guys earlier, you know, that, let me see if I can clear some of this out of here. So I told you guys that we have triplets in our DNA, right? So let me just make up another strand of DNA. Okay. So let's say we had a template strand that looks like this, right? TCGACT. This is our template strand, and we want to build mRNA from this, okay? Now remember, every 3 nucleotides, when you're looking at DNA, is called a triplet. So let's go ahead and build our RNA. So we would have A here, G here, C here. We would have a U here. Remember, we're replacing the T's we use. G and A, okay? So this is our mRNA strand that we have built. And when you look at mRNA, okay, every 3 nucleotides in RNA, instead of calling it a triplet, it's called a codon. Because every 3 nucleotides codes for 1 amino acid. Okay, so each codon codes for 1 amino acid, which, keep in mind that I said amino acids are the building blocks of protein. So we can link these amino acids together, kind of like beads on a string, and we can produce protein from that. Okay. Now, as the mRNA comes out of the nucleus, now it's going to be bound to by a ribosome. So this big red thing here is a ribosome. Okay, it's coming, it sticks to the RNA, and it's going to attach itself initially to what's called a start codon. Now that's going to tell the RNA, the ribosome, where to start, okay, on the strand of mRNA. So it will start on the start codon. And what it does is it kind of sets itself up for these codons to be read. Okay, and as they're read, so let's just say we're working with this one right here, this GCC. Okay, that's our first codon that we want to turn into an amino acid, okay, or use to make an amino acid. So what happens is the ribosome sticks to that codon, okay, that GCC, and then we have what's called a tRNA. Remember I told you guys about that earlier. tRNA is transfer RNA. That's going to be used to carry in amino acids and link them onto a growing protein, okay? So if our codon is GCC, look at these tRNA molecules. Here's one right here. Here's one right here. They kind of look like bent hairpin structures, okay? Each one of them has what's called an anticodon at the base, okay? It has 3 little nucleotides at the base of those. And those anticodons are going to match up with the codons on the mRNA strand, okay? So if our codon is GCC, then our anticodon must be CGG, right? And that tRNA with that anticodon will come and bind to the codon, and then it will add on its little amino acid that it's carrying at the top. So each one of these tRNAs is also carrying an amino acid at the top of itself, and that amino acid gets added on to that growing protein. So here's our growing protein here, okay? Now, after that tRNA adds its amino acid onto our growing protein, then the ribosome will move down 3 nucleotides, okay? So once it moves down those 3, the next 3 that we have are AUA, right? And so what tRNA is going to come in and bind to that? Well, it's going to be one with the anticodon UAU, right? Because that matches up with AUA. And so that tRNA comes in and binds to that binds to that codon, okay? And it has this amino acid attached to it, which it will then add on to our growing protein, okay? Now you will notice that each one of these amino acids is different on each one of these tRNA molecules. So this amino acid, this ALA, for example, okay, is going to be very specific for this anticodon CGG, right? Whereas this ILE is very specific for this UAU, okay? So you don't have to worry about memorizing any of that, or what these specific amino acids are or anything like that. Just know that each tRNA is going to have its own little anticodon attached to it, and the amino acid that's attached to it is going to correspond with that anticodon, okay? And those amino acids are essentially what are going to be used to build the protein that we're trying to build. Now, how do we know what amino acid to add on? Well, we just look at our instructions, right? We look at our mRNA, and that will tell us exactly what amino acid we need to be adding on next to help build our protein. So you guys, let me know if you have any questions after you've watched this video. You can email me at any time, if you have any questions that you think of. But make sure you go through this video, and if anything is confusing about it, make sure you ask me about it before your first test. All right.